John was on my committee, and yesterday first question, John puts his hand up, and just for the split second, I was like, oh, damn, he's going to ask me a question I don't know the answer to. It's just like my PhD committee. It's every student's worst fear. So thanks, John. Really kind. So today's going to be a little bit different for some of you who saw yesterday. I'll be a little shorter because I understand tomorrow is your break. So I want to explore this thread of blow-ground interactions, and I want to dedicate this seminar to Martin Caldwell because he was my advisor here in Logan, and he, at that time, and Jim Richards, his close colleague, were doing really, I think it's still fair to say, the tightest and kind of the cleanest blow-ground competition studies of anybody in the world. He was using dual isotopes, P32 and P33, able to tease apart what individual plants were doing and extracting phosphorus from, and just really, really remarkable work. And so he sort of blessed me and cursed me with an interest in below-ground ecology, which I have continued throughout my career, and it's always difficult, it's hard to get to. So what I'm going to talk about today is a little bit of some of the background, the kinds of things John talked about, and then systems that we've developed over the years to understand these questions. And then I'll also mention Hal Mooney, yesterday, my advisor at Stanford, who kind of helped me broaden my thinking a little bit. Hal was very interested in how the Earth was changing even back then, and so I think those two people really together have helped me think about and define my science. So this seminar is dedicated to Martin. I want to explore roots. This is also unusual in another couple ways. I'll apologize twice, which is always bad. I always tell my students never start your seminar by apologizing, so I'll do it twice. One, I have never given a seminar like this. I'm gonna not focus on. I'll have some new data, but I want to give you sort of maybe more of a broader career perspective on how I think. So I'll do that, and then there's not enough works cited for other people, but there are lots of other scientists who have influenced my work, and I will continue to say that over and over again. So let me jump in, and I want to do a couple of things today. This won't work, so we'll do it the old-fashioned way, or we may not do it at all. Get an early break. So I appear to be frozen. Oh, yes. All right, 3 questions today, with an emphasis on the first 2 in particular. But these are, in general sense, the questions that have guided what has been the longest running thread for my work. And the first one is analogous to canopy structures above ground. How are roots structured below ground, vertically and laterally? I'll focus more today on questions of depth and where plants obtain their resources from, water and nutrients in particular. So the question number 2 there, and how we know that. And there's good work going on today here. USU in that area, and then a little bit on above and below ground traits and how some of these things help us make predictions about where plants may be able to migrate to, or ways that they'll be affected by climate change, droughts, things like that. So these are the 3 questions I'll cover, and we'll come back to this outline a few times as we go along. Well, John was a nice introduction. So I finished my postdoc and started work at my first faculty job, and there were, at that time, no global representations, if you will, of how root biomass was structured below ground. And so one of the first things I did with a number of colleagues was to basically grab all the data I could at that time. This is sort of before meta-analysis was a formal discipline, but so this is not a particularly sophisticated approach, but it's been an approach, been a data set that's still continued to be used today. I had data by biomes. We had data for coarse root biomass, fine root biomass, and sort of a take-home message here. This is depth, and then the sort of the proportion of roots from 0 to 100% left to right. You can kind of see different depth distributions here. No real surprises in the sense the grasses are more shallowly rooted than shrubs and then trees, and a lot of variation within biomes and even important variation within species. But really thinking about how we might represent what's going on below ground in global models Earth system models, ecosystem models, as imperfect as these figures are, they're still used today. So let's think about how and where roots are below ground and how it might influence how the world works and what it looks like. So this is kind of a cartoon. It's actually a graph from a student, Stepan Jobaj. And imagine, so here's a plant. Here's its canopy depth. We're used to thinking a lot about where leaves are structured and how they intercept sunlight and how canopies overlap with each other and shade each other. And things that happen below ground happen exactly the same way, only except for sunlight, they're after water and nitrogen and phosphorus. So imagine a profile, a vertical profile in a nutrient that looks like this. So it might be more concentrated at the surface. There's some depth below which the plant has no ability to access that nutrient because it's below the maximum rooting depth of that species or the ecosystem. So anything that happens below that point is a physical process like leaching or such, which I'll come to in a second. But then imagine at least down to that rooting depth, a plant might be preferentially taking specific nutrients out of that profile, pulling them up to the surface, and then redistributing them through leaf fall back to that surface layer. So the question is how do plants structure vertically elements around the globe, and how would we know and test these kinds of things? So that's what I want to explore for a few minutes, and we'll jump to water. So if you were just interested in a physical process, I think people to that time the time this paper came out, had kind of emphasized the obvious important effects of leaching, particularly for anions that tend to move more. So leaching is going to do this to a profile, right? It's going to take elements and move them downwards through that profile. So that you might get a profile that changes through time to look like this. And then again, the plants will do the opposite thing. They're going to be counterbalancing that flow and moving them to the top if biotic constraints or biotic processes are important relative to physical processes. And that balance between biological and physical processes has really been the underpinning, the intersection that I've been interested in through this work. So how would you test such an idea? Well, one of the first ways we tested this was to go to Argentina. I had some great colleagues in Argentina that I'd met at Stanford, and we went to dune sites in Argentina. They were natural dunes, but that people had afforested with pine trees over recent decades before. So this was a system that was completely abiotic before that time, and then we knew the ages of these pine plantations in that system. So it was like an experiment that you would have liked to have set up, but I could have set up an experiment and gone back in 15, 30, and 55 years, but I'd still be waiting for the end. So we used these profiles. Here's what the profiles looked like from the bare dunes, and there were plenty of bare dunes left that we were able to sample. So this is exchangeable potassium on the top, a plant nutrient, exchangeable sodium on the bottom, both an anion, single charge, relatively even profiles through this distribution. All right, 15 years later, the pine trees are in that system. The profile for sodium looks pretty much the same as it did. The profile for potassium, you're starting to see a nudge of topsoil concentration, right? As the profile starts to get a little bit higher here. 30 years, you're seeing a big concentration, and you're actually seeing the opposite in this. So the trees are excluding sodium, if you will, leaving that in the profile. That sodium is being leached down into the profile. And then by about 55 years, you get exactly the kind of profile and topsoil concentration that you would expect for a process, for an element that was dominated by biological processes instead of those physical processes. And for sodium, you see the opposite. So, Esteban, sort of led this work as a PhD student, and it was really a nice tight experiment to show the importance of these biological processes. And then he went on to think about this globally. Now, we didn't really have an experiment to do this globally, but we could look at the relative profiles. So what we did was to take thousands of profiles available, organized by scientists in the Netherlands, the WISE database. Neil Badges and people have done the best job of assembling these kinds of data, and that's still true today. But you can imagine what we call the topsoil concentration factor for each of these elements here. So a value of 0.2 means you've got 1/5 of the element in the top 20 centimeters, exactly what you would have if the distribution were even throughout there. And anything above this line is a topsoil concentration, and anything below this line is a heavy tail deeper down, what you'd expect from leaching. And so you see phosphorus is the element with the highest topsoil concentration globally. So more than 40% of P is in the top 20 centimeters of soil, of all the P that's contained in the top meter. The organic elements, carbon and nitrogen, are also highly concentrated, and that's a little bit different process. And there you have litter and plant fall building up at the surface. Potassium then here, the next here, and then you start to stretch out. Calcium down here, sodium way down here, sulfate to the right. What just happened? So again, those are now anions that are moving, and chlorine too. So globally, there are plenty of processes going on here, but globally there's a strong biological influence on the distribution of these elements around the world. Okay, so those kinds of ideas are based in part on distributions of roots, particularly in the top couple of meters. I've always been intrigued by the exceptions. And the exceptions are the cases where you read an old study from the 1920s and somebody finds a mesquite root down 50 meters underground. Right? And so it's very, very hard for us to understand how common that is, whether it's very important, how much water, what are they getting down there? There's probably water in a dry system, but how could you quantify what plants get from these deep roots? And they're not very common. So this is a study that Jochen Schenk led about 15 years ago, and it was a different kind of database work, much bigger. But these are not sort of where roots are just in terms of the biomass. These are where the deepest roots are in given systems. And this is now individual plants rather than ecosystems. So these are observations from our work, but also from the literature. And you have here, well, now this has gone around, wrapped around, but grasses, shrubs, and forbs, and then annuals and succulents are very shallow. And over here you get towards the trees and shrubs in particular. And so these are the means, the arithmetic means. So typically, in most systems, you've got a few plants, you have individuals that are down at least a few meters, but then you've got, in some of these cases when they're down really, really far, tens of meters. And it's, again, extremely hard to understand, how to predict that, and then also what those mean functionally. So one of the things we did, followed up then, was to try and take all the data that we had for where deep rooting occurs around the world and try and build a model around to predict those, at least in a general sense. And we tried to do that primarily based on what you would expect, soil characteristics of texture, whether it's sandy or clay, how quickly water moves down through that system. We looked at climate, so rainfall. You might expect deeper roots in drier systems, but if a system is really dry and the water never gets very deep, then you have plants like cacti whose roots are very, very shallowly distributed, for instance. There are aspects we expected of seasonality to be important. So if you have a system like the Great Basin here in Utah, where you have a lot of rainfall in one season, drier summers perhaps, as we have in California, then for those trees to maintain leaf area, they're probably going to have to be down, extracting water from areas of the profile after the top layers have been exploited fully during summer. So when you have a long gap in rainfall, you would expect deeper roots to occur as well. And where you have higher demand, higher evapotranspiration, you might also expect to have deeper roots. And so all these things turned out to be true in our model. Soil texture also mattered. But so we got a picture that looks something like this, and the sort of the redder you are on this figure, the higher the probability of the system having deeper roots. And you see some things that you would expect. You don't see a lot of deep roots smack dab in the middle of the Amazon where it's very wet, but you see them around the Cerrado, the savanna system, seasonally dry systems. You see them across the Sahel and other areas, savanna-based systems, thorn scrub through the eastern U.S. So this model predicted pretty well where at least the observations had shown that such plants existed and the deep roots existed. So I've also long been interested in taking sort of the data that we can find, such as they are, and trying to build predictive frameworks that we can then run out and test. If there's a place that says there should be really, really deep roots and nobody's ever been there, we'll go there and look for them and see if we can find them. Sometimes you do, sometimes you don't. In terms of new data sources, we continue to do this today. I have a grad student, Sher Singh Tumber Davila, he's mining the new literature. He found this, he and Jochen together, we still work together, there's this amazing data set, and this is really old, even older than I am. This is like 1930s, 1940s in Europe. That's a tree. That's a root system, and they have painstakingly extracted that system, used wires to hold those roots in place, and then gone down in that system. So that's sort of the 3D picture of the root system held in place by wires. So with these kinds of data, we can look at the relative abundance of roots in shallow. We can look at the relationship of lateral width. So how much overlap. In most tree systems, the roots extend beyond the canopy edge. So they're competing out there with other individuals. They're often wider than they are deep, although not always. And so we have a couple thousands of these kinds of observations that Schersing is digitizing and using, that we compare canopy structure above ground. So how does the relationship change of how wide a tree is in its leaf area, and how wide a tree is in its root structure, and how deep the roots are versus how tall the canopy is. And so these volumetric relationships are what he's looking at right now and what we can learn from that. We have, and I'll show you a few examples now of some of the constraints below ground. So, we know the roots can grow very deeply. They don't always, and in most cases they don't. Some of the things that can both be a resource and a constraint include groundwater. This is work that we did led by Yingfan Reinfelter. She's at Rutgers, just a colleague. So this is kind of maybe a geomorphologist type picture. So you can imagine a tree here. There's an infiltration depth. This is how deep the rainfall goes. Here's the root distribution. And then there might be groundwater way down here. That groundwater is not accessible to that tree. But you move down slope, and at some point, the groundwater and the rooting depth intersect. And maybe even where that's deep, the plant has a deeper root system to access that groundwater. Then you get down here, when the groundwater is actually a constraint. It keeps the root system from going very deeply, because that groundwater is usually low in oxygen availability. And we'll talk about that in a few minutes. So, in this paper, we used more than data for 1,000 species, several thousand locations to look at relationships of groundwater depth and rooting depths as well. And this was based on Ying's paper earlier, not that I was not involved with, but she produced the first global map of groundwater depth. And it's a static map, which in and of itself is not the way groundwater works. Groundwater fluctuates, but there was not a gridded product of groundwater depth before that time. And she's continued to update that. All right, so we built a different kind of model in this case. So we took observations of, it's really more of a hydrologic focus. So what we call an inverse model. We took 10 years of data, of climate data, looked at the texture of the soil. So if you're a sandy soil, then the water percolates more deeply, more quickly into the ground than in a clayey soil. And we looked at, say, over a 10-year period, what's the driest, what's the deepest plant systems would have to go to survive, to get enough water. So if you have lots of rainfall regularly, that root system might never have to be below half a meter or a meter. But if you have 1 year that's really dry and a long seasonal period, you push that depth necessary to obtain water deeper and deeper. And so what we got from this picture was a sort of a distribution. It's a long-tail distribution, but most of the systems are up here in this 4 meter range. Based on this inverse model, the data sets, the observations are in the points there. And then, then there are these other systems that are farther out. And then in a spatial way, this is the prediction of rooting depth, kind of like the Schenck map I showed you a few minutes ago, but more sophisticated and done using a completely different set of data now. This is hydrology and water-based rather than root observation-based. So blue is shallow, and blue is where you would expect boreal forest. It's cold, relatively low leaf area, little evaporation. Blue in the Congo, in the Amazon, in Indonesia and Borneo. And then the reds are the deepest, and you see them in really quite similar areas to where I had them on the previous map, in the Sahel, over here, the Sado, savanna systems, parts of Australia and things like that. So this model seems to work pretty well, and we're trying to update this and then also test it with some new data sets. And then one One example of the constraints now on rooting depth is a graph here, finally, that I'll show from that paper. On the right is the distribution of observed rooting depth and the depth of groundwater. And on the left is the distribution of bedrock and rooting depths. So what you see here is that for groundwater is actually a really, really tight constraint. And that below about a meter, there are some plants who are actually growing well into the groundwater. But once you're down a few meters, then because that groundwater is almost always low oxygen, very low oxygen, the plants don't grow into that. They stop, and they probably stop, and they're perfectly happy to extract the water that they can get. But it really does act as a barrier. So if we had good maps of groundwater depth, we can use that to constrain the sort of the zone of biological activity. Bedrock's a little more interesting. Bedrock constrains depth on average, but then there are many examples of plants that can grow into rock. And I'm going to show you some examples of that in work that we've done. So we're thinking, I think about this both from the standpoint of how far plants can grow, but it's not just about how plants doing this. It's about the physical constraints that come up from underneath them. All right, so let's look at the second question. What depths do plants obtain water nutrients from, and how can we know? So that'll be our next topic. I wanted to talk about a couple of systems. I'll start with some work in Argentina, again in the spirit of doing sort of the tightest comparisons that we can make to test ideas. And then I want to go to Central Texas to explore our cave system, and then some recent work on mortality and droughts and how water availability influences the outcome of those experiments. So we've been very long interested in vegetation change, so processes like woody plant encroachment that's happening in the western United States, afforestation in Argentina, which is what you see here, provincial incentives for people to go in and plant pines, plant eucalypts where there never were trees in the past. And so we use these kinds of human manipulations as our experiments, if you will, if we can identify the species and the history of those, the age and history of those stands, whether or not they were fertilized and managed, things like that. And then you see a woody encroached system like this in Texas. So let's look at Argentina. So these are a number of study sites that we worked at for a lot of years around the Buenos Aires area, in Uruguay. And this is an area where that was treeless. So this is one of those unusual places in the world where there's plenty of rainfall to support trees, but as far as they can tell, except with some exceptions along riparian zones, there weren't trees. And there are historical photos going back 100, even 150 years, showing these treeless plains, and then the cowboys, chasing after rheas with their bolos, on the horses, and there's nary a tree in sight. But people in all this area have gone in and planted trees, and the trees have done just fine. So even today, really, ecologists down there don't understand really why there weren't trees. It's not a fire constraint. It doesn't appear to be a disturbance constraint, but there was something that kept them from surviving. But people have come in and altered that. So let's look at one such example. You could imagine taking a treeless pampas grassland like this. Now this is a system that's quite wet. It's very, very flat. So water does not move laterally very quickly. This is why when they have a heavy rainfall year, you see pictures of the area within 100 miles of Buenos Aires completely flooded. And provinces fight to dig channels and get the water out of their province and somewhere downstream to the next province because the gradient is so low. So there's groundwater in these systems. There are grasses here, and then you come in and let's say you put a eucalyptus plantation or a pine plantation in there in the middle. And you can imagine these systems have very high leaf area, higher than the grasslands, which we have measured. They're deeper rooted than the grasslands, so you might expect the trees to use that groundwater and maybe to deepen the groundwater, depending on how quickly it can be replenished laterally. You might expect them to alter the salinity of that groundwater. If they're taking the groundwater out, they might be preferentially leaving behind salts. And then in this system, drinking water comes from a film of fresh water that occurs at about 5 meters down. So this is what houses use in that area for their use in homes. So this is an experiment. That's a cartoon, but let's look at an example of how this works and what trees, in this case eucalypts, do below ground. So here's a a plot of planted trees. This plot's about I think 40 years old. You got croplands, grasslands around here, grasslands down here, some crops there, and such. And so I want to explore a transect across this this plot. So here is these are boreholes. These plots. So we have wells that we can go down and sample the depth and the salinity of the water in the system. We can measure the salinity. There's a windmill, a couple of windmills in the system, so we get very, very deep estimates of the salinity of the groundwater. And then we can run transects using geoelectric profiles. So these are instruments that measure the conductivity or the resistivity of what's occurring below them in the soil. So they propagate a signal, and that signal moves more quickly if there are a lot of salts in the water. So salt water conducts much more quickly than fresh water does. So by using how quickly that wave propagates, you get a sense of what the salinity is, and then by running these transects, you get a more three-dimensional picture. So these are the kind of data sets that we collected, and these are the kinds of results that you see. So here in this case is one transect that runs from the top in the grassland down into the eucalypt plantation, left to right, grassland, then the plantation on the right, distant to the edge. Here's the boundary. Here's the electrical conductivity of the water. And here's the boundary there, and you see the jump up in the salinity, just as you would predict from the cartoon that I showed you before, or a quantitative model. So that occurred across all of the plantation transect boundaries we showed, and basically in all the systems that we studied there. So red is fresh, blue is more saline. Grassland here, grassland here, grassland here, and then plantation, plantation, plantation. You see the absence, the disappearance of that freshwater lens in red under the trees. So they've used that fresh water up. And you see higher salinity, which is some upwelling of deeper water, but is also the exclusion of salts, preferentially by the trees too. So this is a biological process that has changed the depth of the groundwater and the salinity, the quality of that groundwater too, in a few decades. Now, if you want to look more broadly, you might ask, well, that was one species. Another thing we did was to say, what's the maximum salinity tolerance that different trees might have? And this was work by Marcelo Noceto, another Argentine colleague. And in this case, we went into the literature for physiological experiments, and trees have different tolerances to salts. So you can imagine looking at different species within a genus, like eucalyptus here. There are cedars and poplars and such. And there are trees that are more tolerant and less tolerant of salinity. So we then took those tolerances and went to plots where those trees had been planted in Argentina and Uruguay and looked at the salinity of the groundwater that was found under them after they'd been established for at least a few decades. And we saw this relationship here. You see, so not only do they alter the salinity of that groundwater, but they alter it to the extent they're able to use that water, and then no more. Right? So once they salinize the groundwater to their tolerance, or higher, they stop being able to use that groundwater, and the system stabilizes at that level of salinity. So based on the tree species that were there, we could predict what the salinity of the water was under them, just based on those tolerances. Let me finish up in Texas. Now, it's been a while. This actually was the president long ago. That's George W. Bush clearing cedar from his ranch in Texas. This is a woody encroached system there, and I'm going to talk about this area in central Texas, one of my favorite places on the planet. That's the hill country in central Texas. This is a karst system. So now instead of groundwater, we're thinking about bedrock. There's a karst system. By definition, karst is a fractured limestone. It's very common here. It's very common in the Yucatan, a number of places around the world, Australia. So very shallow groundwater, 10, 20 centimeters of soil, like a film, over groundwater, over bedrock, and but trees that are able to survive on this system. And this is an example of a system where you basically have to be able to grow into bedrock, or you don't survive, because there isn't enough water that's held in that topsoil layer, that film, over a hot summer like Central Texas has. So one of the things, best project we ever had in my lab, we got to cave to study water use for plant uptake. So imagine, let's start with a figure that's like this. So imagine you have 3 tree species along the top, and you have a cave Now, this cave might be 5 meters down, might be 10 meters down. I'm standing in your way, aren't I? 25 meters down. All right, so you can imagine thinking, just as a tree is tall, a tree has to have different depths underground. And you can imagine moving this cave up and down. And if you can go into the cave and find roots, they might belong to any one of these 3 trees. But as that cave deepens, the shallower rooted trees start to drop out. Until you get to a place where there's only one species left. And at some point, of course, there's no tree species down there. So that was the spirit of this study. First of all, we found a whole bunch of caves at different depths all over the central Texas area. And I had tremendous help from the Spelunking Society at UT Austin there in doing this work. All right, so we went down there, found lots of roots in these caves. Sometimes they were fine roots, sometimes they were tap roots. And I'll show you some pictures of these things. So then you're down there, then you have to figure out who's down there. Let's say you see roots, but you don't know whose they are. And it's not 3 trees at the top, it might be dozens of species. So we tried labeling, we put labels and roots, and it was hard to do. And finally, we ended up working with a colleague, Randy Linder, and developed the first molecular tools to identify them first as species, and there was good work being done here at the time in this area too, first to species and then to individuals. So now imagine after enough years in this system, we have maps like this all over the hill country with caves where we know who's at the top, we know whose root it is down here. I can instrument this root with a sap flow gauge and measure water uptake in real time, 24 hours a day, 365 days a year. I can take shallow roots up here at the top, trace them back to the tree, instrument those, so you can start to get a relative sense of where water's coming from, different times of the year, when the shallow soil's wet, when you're in the middle of a drought, winter, summer, things like that. Plus, we got to go caving, so everybody loved this. Everybody loved this project. So first of all, we found a couple of things. We found that all the tree species in the system were at least 5 meters deep. So you didn't find every tree in every cave, but as you went around the system, you could find all of the oak species. You found the juniper. You found Diospora, basically, the dominant species there. The deeper you went, the oaks tended to be deeper. And then as you got really deep, down below about 20 meters, there was only one species we ever found. That species was the only evergreen oak in the system, Quercus fusiformis. So the only tree species, the only oak, the species that had to maintain its leaf canopy 365 days a year, was down the deepest, essentially as an insurance policy, to have enough water to keep the leaves on the trees. I know we confirmed this is a 25-meter deep underground stream that we worked at in the cave. So we were able to confirm isotopically. This is a deuterium study. Here you have surface soil is here. You have shallow species up in here. Here's the signature of the stream, and here's the signature of the water that was in the branches of the oak. So, and through other approaches, through sap flow and such, we had direct evidence that the trees were using that water. So through sap flow gauges, this was work that people like Andrew McElroone did. So here's a columnar taproot. That's a juniper taproot. That's a relatively shallow cave, 6-7 meters underground. You've got a juniper there in the middle. You've got 2 oak roots weaving its way around that column, moving down, the thin ones. These are roots that we have instrumented with sap flow gauges to measure the water flow through them. And then here's an example of, this is not like Carlsbad Caverns. These are kind of little cracks in the ground, and you have to be willing to get a little dirty. All right, how does this information help us? This graph's a little complicated, but let me work you through it. So here's, first of all, rainfall, all right, and then over here is the soil water holding capacity. So these are 2 rainfall events, 1 here and then 2 here. Dry here, wet, a little bit of drying, wet again, then drying curve. All right, so let's start with, so these are different roots. So this is a deep root here, lateral root here, same tree, this is an oak, and a lateral root here. All right, so look what the deep root's doing. Now that root's, in this particular case, that root's about 7 meters underground. So you see a daily fluctuation in flow through that root. That's not surprising. That's a transpirational stream, but you see that root being quite important. It rains at the surface, the deep root basically shuts down. Right? The shallow root does the opposite. It's here, supplying almost no water. It rains, and then it becomes a source of water to the tree. And then another one here, you actually see a lateral root that was providing water, and then it's actually it's sinking water down into it's leaking water out. This process was discovered by Jim Richards and Martin Caldwell-Hero. They called hydraulic lift or hydraulic redistribution, where water can actually move through the tree based on a water potential gradient. So if you have dry soil at the top and wet soil underneath, and the water potential gradient is high enough, it will overcome gravity and move upwards through the root system and leak out into the surface. It's not a huge flow like a bathtub, but it's measurable on a daily fluctuation. So these kinds of interactions are what you're seeing in this root. So the deep root was only important in these dry conditions, but when it was really, really dry, that single deep root was providing almost a third of the water that tree was using. And that's very hard to estimate in a model. It's very hard to estimate in terms of biomass. It's relatively low biomass, but that's the survival mechanism. So in this system, the trees are, in my view, are playing the lottery, and they're all growing deep roots. They're probing, and probably most of the time they don't find water, and those trees don't survive. But the trees that find the water in basins that pool or whatever it is are able to survive through the dry conditions, and only those trees that do make it. At least that's my model for it. So let me finish just with one final and newer work, some examples of the same system now in Central Texas, but following the consequences of a drought in that system. So I'll tie back into a little bit of climate change from yesterday. This was the 2011 drought that hit Texas so severely. Based on the tree ring record, this was the most severe drought that they'd had. The 1950s were drier, but this drought was a lot hotter than the 1950s. So this was particularly dry and particularly hot. And this is the drought monitor from, as you can see, September of 2011. And this is what you saw, within a year. You saw that patchy mosaic of death and mortality, and then live trees, just as you see, here throughout the Intermountain West. So we wanted to understand what might be causing that mortality and also the extent of that mortality. So this is work done by a grad student, Amanda Schwantes, postdoc, and several other folks in the lab. So first of all, how would we assess mortality across this area in Central Texas and then look at the whole state as a whole? Well, we did this through a 3-tiered sampling method. We had about 20 plots of transects that we measured on the ground in different systems across the state. So we went out there in the field, put down random transects, and then measured for many, many hundreds of meters the number of trees, the number of live trees, the number of dead trees, directly over the transect, and in some cases, belt transects. So we had ground-based measurements. We used high-resolution aircraft photographs that the state had taken before and after to be able to scale those ground-based estimates up and make sure that what we were seeing or thought we were seeing based on the photographs was actually the same thing as mortality we observed on the ground. And then we used those photos to calibrate a remote sensing approach using MODIS and Landsat to then look at the whole state and identify the pixels that had changed. So we did that. This is a figure of the state of Texas. Wherever you see a dark pixel, here is where there was higher mortality in this system. So in this case, at least 25% of the tree canopy cover in that pixel died. Right? The mortality wasn't random. So you can see there was a lot in South Texas, a lot in the hill country. So greatest canopy loss, post oak woodlands, pinyon juniper shrublands, like I showed you in that earlier Central Texas system, ash juniper woodlands. We lost some turkey oaks. We lost a lot of junipers, which were interesting. We did a lot of work on kind of ecophysiology of drought tolerance, gas exchange. The junipers are the most drought tolerant species in this system, and yet they showed a lot of mortality. So it isn't just about their anatomy and their physiological characteristics. They died in a lot of places where they had encroached into also. So they were in sort of marginal habitats to start with. And one of the things that correlated with this mortality, so even though I talked about how important the bedrock depth is for water, the surface fill matters, and this mortality was correlated strongly with the shallowest soils. So where you had the shallowest film in the surface is also where we saw the most mortality in the system. Right, so it's an example of how, think about how one drought event can change the canopy cover of an entire state like that. And we're starting to see these kinds of changes today across especially the western U.S. And finally, we can say, well, what does an event like that tell us about the future, and how do we know? This is a figure that shows, in this case, so this is the percent forested area impacted. This is the deviation of annual precipitation, so minus 20% from the mean, 40%. And you see in this case that once a pixel was below sort of 40% of the mean, 40% reduction in precip, you started to see a lot more mortality. You saw the same kind of threshold for the temperature anomaly. Stop it. Here. So in this case, temperature anomaly, a degree C warmer than average, 1.5 C. Boom, you get out here in sort of the 2 degrees C and more hotter. You again have a stronger mortality effect. And you had an effect on the vapor pressure gradient too here. Same sort of threshold, about 2/3 of a kilopascal. All right, so there was mortality across the whole system, but there were these thresholds. So here we were in 2011, and you see here now these are different models that suggest what the climate will be like in the future in this part of the country. This is a sort of a moderate climate change scenario. This is a stronger business as usual hotter scenario. And you see these thresholds being passed from what were exceptional events, or an exceptional event like this one, into being not necessarily common or every year, but much, much more frequent. And so what is exceptional, what is becoming exceptional and is exceptional today will be much more common as we move sort of 25, 50, 75 years from now. So it's really interesting to think about what will happen, how important will these thresholds be, to what extent will these trees have genetic changes and be able to change, and to what extent will species shift through this system. But the system is changing today, we're already seeing its effects, and what's projected to come suggests that it'll change a lot more. Based on this work, at least, we would expect a lot lower canopy cover, at least for some of these species, than we have today. So I'm going to stop there, and again, my goal today was to, I hope to show why below ground processes interest me, why in many ways I think they're harder than. That we have to work harder to get below ground, to design systems and experiments that we can test things in a really direct way, or as a direct a way as possible, but also why they're important and why I need to couple what's happening below ground with what we observe from the surface. As I said, I would never have started this thread of work without having worked in Martin Caldwell's lab here, so that was the reason I dedicated the talk to him, and thanks for listening, and have a good break. Any questions? Mm-hmm.
So you talk about the bedrock constraint root and the soil depth, and also some sedimentary deposits contains a lot of soil organic carbon. So I was wondering if is there any significance to study different bedrocks, how they contribute to carbon stock, like estimate the global phosphorate bedrock, their contributions to soil carbon stock based on some field experiments or some models?
So let me make sure, first of all, the question was about different kinds of bedrock and carbon stocks, I guess, associated with the bedrock. Is that correct? So I think for water holding capacity, the part below ground that goes into bedrock can be important for water. There's some interesting work recently that different kinds of bedrock may actually supply nutrients, even nitrogen to some extent in some cases. I don't think, if I understand your question correctly, I don't think that the bedrock itself is an important source of organic matter. I mean, there's some organic matter you see in cracks. We see organic matter that's flushed into these caves. There are bats that deposit guano, so they're important sources of nitrogen in these caves. But as a pool of carbon, there's just not really very much volume that's organic matter in a bedrock system. So if that's the spirit of your question, I don't think it's that important. I don't worry about these sort of bedrock systems for estimating the total amount of organic carbon, for instance. I think that basically stops with the bedrock, but the water flow doesn't stop. So I hope that answered your question, if I understood it correctly. Dick?
So Rob, that last study would suggest that you talked a little bit about afforestation and reforestation yesterday, that we need to very carefully consider what species are used in that and maybe try and predict ones that would survive in the warmer.
Yeah,
Is that being done?
absolutely. I think it is being done. I know the Forest Service in places is thinking about that. The commercial forestry companies are experimenting with moving different kinds of trees further north, and even the wineries and where the vintners are moving north and experimenting with different varieties and things like that. So I think I think it's more in sort of natural systems. I mean, there's a lot of work done, as you know, on climate envelopes, work being done here on how quickly species will be able to migrate, whether they'll be able to keep up with climate as it changes and such. So it is something that people understand. I don't think it's something we're using enough, really. But I don't know if anybody in the audience has a comment to offer, or an idea to offer in that thread, or you, Dick, for that matter.
Thanks for the talk. I was kind of wondering from your Texas example, what stopped the other trees from reaching the water level? Was there a kind of trade-off like that happened between investing in roots or?
Yeah, that's a great question. I mean, clearly it's not free. I mean, there's a carbon cost to having deep roots, and of course, we don't know. We never know the trees that get stopped, right? I mean, there are caves that you go in and you don't see roots, especially at depth. But even in some shallow caves, you'll get to them and you won't be able to find anything. You don't know if they're there and you don't find them, or if you don't know that there's some tighter bedrock or a lack of cracks above that doesn't allow. There has to be a fissure or something that allows the roots to grow through. They can't, once they can get in even a small crack, there's work that shows that the acids that in them can provide some dissolving of that limestone, but they can't create over the lifetime of a root in a tree, they don't create their own channels de novo. So I don't know what stops them. We just know sort of the ones that make it through. So it's a great question, but there has been some modeling work, and not work that we've done, but trying to understand the cost of the carbon associated with it. But I think to do that properly, you need to know something about how many times a tree tries and fails, as opposed to it tries and succeeds. So if the cost is high, but that deep taproot keeps you alive, then almost whatever you spend on it is worth it. But if you've had to grow 100, if you've tried to grow 100 deep roots and one of them makes it, then the carbon cost is even higher, and it's probably not 100, but it's not 1. And I don't know exactly what the number is and how often they probe and try. But it's interesting on it, just finally, and I'll stop that answer, but we've worked on a number of underground streams, and once so we go through these streams underground, and you can see tree roots in them, and once you know and kind of get a sense where the streams are underground, and you get to the surface, you can see the trees at the surface that are, they're taller, there are more of them in these systems. You can actually see the effect of the surface on the density and the size of the trees that are able to tap into those underground streams. But most of what we do isn't a stream, of course. Those are exceptional cases. But other questions? Mm-hmm.
Yesterday we were talking a bit about negative carbon emissions, and within the realm of people thinking about how to draw carbon from the atmosphere down into the soils, there's kind of two ways of doing that. One is in biomass, obviously, but the other is in soil organic carbon. And there's been a lot of attention on the relationship in between what looks like the food web up here, but kind of the biology, the mycorrhiza, that sort of thing, and the ability of that to kind of help roots bring carbon exudates down into the soil. Mm-hmm. And I'm curious if your research has any implications for that sort of thinking in terms of where kind of the edge of that field of science is going.
Well, I hope it does. I don't know about these bedrock type studies, but if we think about the vertical distribution of biomass, where I showed you in the first couple slides that I illustrated, if we think about different functional types of plants, and then different species too that allocate a different proportion of their biomass below ground, I think it does. There are many studies, including some of our work, that shows that a piece of root carbon, for instance, that dies in situ is 4, 5, 6 times more likely to end up as soil organic matter as litter and organic material that falls from the surface, say in leaves, then has to work its way through a litter layer and the decomposition that happens there, and then has to get down into the organic matter. So the best way, the quickest way that we can build up organic matter in soils is actually to have relatively high allocation from grasses are good at it, certain tree species are good at it. So by species selection and understanding their allocation patterns, I think we can speed up the process of stabilization of organic matter. Anybody else? All right, thank you.